Hello guys, today I'm going to discuss about the topic southern blotting. First of all, we will define the blotting. What is blotting? Blots are a technique for transferring DNA, RNA and protein into a carriers so they can be separated and often follow the use of electrophoresis. The southern blot is used for transferring DNA and the northern blot are for RNA and the western blot for the proteins. If you're talking about the types of blotting, that is southern blot, northern and western blot. So my topic is about the southern blotting. It is about to use to detect DNA molecule. The first step in southern blotting is the digestion of DNA molecule. First of all, we cleavage with one or more restricted enzymes into restricted fragments. After that, we transfer that very fragments into a gross gel electrophoresis. The third step is about the gel electrophoresis. Through the restricted enzymes, the fragments of DNA molecule is now passed through the electrophoresis. In electrophoresis, a current is used, electrolytic solution is used in that very electrophoresis. According to the density and size of DNA fragments, they were separated into different places. After that very fragments separated in electrophoresis and the gel that are transferred into the membrane. If you're talking about the membrane, that is consists of transfer buffer solution, lead foam, sarin web, ointment paper, gel, and membrane is over the ointment paper, gel, and the paper towel stacks, and weight over the paper towel stacks, so that the fragments of DNA can easily bind and permanently bind with the membrane. After the transfer of DNA fragments from gel into the membrane, the second step is about the hybridization. Hybridization is remove pre-hybridization solution and an hybridized solution, add probes so they hybridized with the DNA fragments, hybridized overnight and appropriate temperature. Now we overview the whole process about the southern blotting. That is, in the first step, we take the blood sample, and in the second step, DNA is extracted from the blood cells, and the third one is DNA is cut into fragments by restricted enzymes, and the fourth one is the DNA fragments are separated in bands during electrophoresis in an aqueous gel. The fifth one is the DNA band pattern in the gel is transferred to a nylon membrane by technique known as the southern blot. And the sixth one is the radioactive DNA probe is prepared, and the next one is the DNA probe binds to the specific DNA sequences on the membrane. And the eighth one is the excess DNA probes is washed out that are in the solution. And the next one is, at this stage, the radioactivity probe is bound to the DNA pattern on the membrane. And the second last one is the X-ray film is placed next to the membrane to detect the radioactive pattern. And the last step, the X-ray film is developed to make visible the pattern of bands which are known as DNA fingerprints. Now we are going to discuss about some applications of that very southern blotting. That is, the number one is presence or absence of gene. The second one is count the number of genes, and that is used in reflips and Flips too. Thank you so much for listening.